It's gonna be about insects under the magnifying glass, how collections help answer biological questions.
Here's an overview of the diversity of collection of insect specimens that you can encounter in the collection. Back in 2021, I had this bunch of specimens I wanted to extract label data from. And back then, we realized by taking a picture, you could efficiently extract the data from the images. And so we developed, following years, a pipeline to make use of the high performance of OCR, which was quite high for printed labels, but performed really low for handwritten labels. So the idea that we developed here in the pipeline is just to separate a handwritten from a typed label, and so proceed them differently. So handwritten labels are still typed by hand, while printed labels are processed over the OCR. And also the main added value of a pipeline is that after this OCR is performed, we just process through clustering of these labels, since there are many redundant labels in collections. So for instance, when someone has been collected at one spot, he collected, for instance, a few hundreds of insects. And so all of these insects bear the exact same label. So I will go shortly on this, but this is a 2D representation of the label output that we had. And so we can see that they form like several clusters of similar labels. And this way, by only checking manually the two most distant labels in each cluster, you can infer the information of the whole cluster. Take-home message for a pipeline here is that you can automate this way the reading of 2/3 of the labels. So now to show you 3 examples of how collection specimens are used in research. So first, I'll start with the systematic research. So collections serve as a base to describe the name diversity out there. And well, to discover new species, for instance, necessarily you need to go on the field on expeditions. And here, while sitting in Berlin, I just investigated the material we had from the Philippines here. And this way could highlight that of the 64 species we had, 32 were actually new. So half of them were new and undescribed. This is the kind of research that we can do with a collection specimens, is just do what we call like ground research, which is mapping and naming the diversity that is out there, especially in the tropics, where most of it is still undescribed. So that is one of the first options to use collection. Another example of the way collection specimen has been used is that's by a colleague from the US who traveled around different museum collections and measured the wing length of the famous monarch butterfly, this butterfly who does big migration from the US down to Mexico. And he could this way highlight, by measuring a wing length of specimens from all over the world, a trend that when this monarch butterfly colonize and settle oceanic islands like the Azores in the Atlantic or Pacific islands, they tend to reduce their wing length because the cost of having big wing length means that it will more easily be blown away by wind and then die this way. And so there is a trend in butterflies colonizing new islands to have their wing length reduced. And here, further example that we have, researcher from the UK visiting the collection last year, and this research team wanted to investigate the genomics behind the black form of the peppered moth. So you might all remember, some of us had at school, where you have this peppered moth that is normally white and grayish. And during the Industrial Revolution, phenomena started in UK, so that the tree kind of became black because of the coal mining industry. And so a form of this peppered moth appeared that was blackish, and that will, of course, mimicry better on trees. And this form spread quite rapidly because, of course, suddenly the white peppered moth were much more visible to birds, and then selection played against them, and they were more easily eaten by birds. And this phenomenon, this black form, spread then at the end of the 19th century over Europe. And so this researcher was here last year, and he sampled a few specimens of our collection. That's just the perfect research example that shows and acknowledges the richness of our collection, because we have many specimens dating back to 200 years. And this way we are able to investigate this kind of rapid evolutionary mechanism that spread all over Europe by investigating historic samples from a different location. And yeah, nowadays with new techniques, we're still able to get whole genomes that are a 200 years old specimen that has been sitting in collection for 200 years.